So we'll dedicate 3 lectures to gene expression regulation, which I like to abbreviate GER for short. Today will be about GER in bacteria and prokaryotes, and then next week in eukaryotes. And you know the drill by now. I like always to start with a sort of an open-ended question. We know that in multicellular organisms, animals, plants, all cells have the same DNA. But then it begs the question, why aren't all the cells the same? In any of us, we have muscle cells, neurons, intestinal cells, and so forth. But they all have the same DNA. So how come they're not all the same?
Maybe the structure of the cell.
Okay, so what you're saying is different proteins will somehow affect the structure and function of cells. Totally agree, right? And how do you get different proteins in a cell if they all have the same DNA? Why don't they all have the same proteins?
Like different parts of the DNA is being used?
Okay, so different parts, so different usage of DNA. So I'm going to write like this: different cells use different DNAs or DNA regions, parts. We do get to what you were suggesting before. By using different regions of DNA, you make different proteins, and those ultimately will carry out different functions in cells, which is what makes cells different, right? Any other ideas? In which other ways might cells be different despite the fact that they have all the same DNA? Yeah. Cell differentiation, okay, and what does that mean?
They become more specialized when they differentiate, so it's different.
Perfect. So that's a sort of a functional description of what cell differentiation is from the cell perspective. Cell differentiation means that cells are very different and they become very specialized, right? And that's all correct. What does that look like, though, from DNA's perspective? How do we tie the fact that these cells differentiate and become different and they specialize with the fact that they carry the same DNA as all of the other cells in that body?
I guess from the last lecture, some of the DNA are active and some are inactive.
Silent, I like that word. We're going to use that a lot. Yeah, so not only different cells use different DNA regions, but also different DNA regions, and we'll explore this a little bit more next week. So different DNA regions differ in, and I'm going to change the color here, accessibility. So not only is it a different use of DNA, but there is also a series of processes by which different cell types have regions of DNA that are completely inaccessible to them. And that's how they achieve this degree of specialization. There is a point in the life of cells in multicellular organisms at which, even if they wanted to express certain genes, they can't, because that DNA became inaccessible to them. Okay? When I bring up these sort of same questions, some of you may have taken the previous, the prereq to this class 107 with me may remember that I start with how are different cells in a multicellular different? And the answer that I often get is because they have different DNA. By now I'm assuming I kind of went straight with all cells have the same DNA. I assume that you guys knew that, but probably not bad idea to reinforce that idea, that concept that all cells in a multicellular organism have the same DNA. They use it differently, okay? They may use certain regions that other cells don't, and vice versa. That's relatively straightforward, right? Different cells use DNA differently, and therefore they become different because they express different proteins, they have different functions. Now, a slightly less obvious question. Bacterial cells are all the same. It's not like in a multicellular organism that different cell types do different things. Bacterial cells, they all do the same. Now, we accept that, and at the same time, I should tell you that not all bacteria, let me phrase it this way. One might expect then that all bacteria express the same genes. They're the same, right? So it kind of follows that if all bacteria are the same, then they're all expressing the same genes. And yet, it is a fact that they don't. So why wouldn't bacterial cells express all their genes all the time? Right? Why would even bacteria that have only one job to do, why would they choose to express certain genes at certain times and not others? That becomes a little less obvious. In a multicellular organism, one might say, well, a muscle cell doesn't need any of those neuronal genes, so it won't use them. An intestinal cell won't need any of those pancreatic genes, so it won't use them. But bacteria, why? Like they're all the same. Why won't they use all the genes all the time? I'm going to give you another minute to think about that one. That's a little trickier, and see what you come up with. Why wouldn't bacteria express all their genes all the time? What might be the advantage of turning certain genes off, though?
So they could save it for later, just.
Okay, so economy. Supposed to write. There you go. Economy. Yes.
Like they would be conserving energy too.
Economy, conservation of energy, right? Like the idea that it's more economical to not waste resources in something that you don't need right away, and anything that saves resources might be evolutionarily advantageous. So, yeah, okay. What else?
Can it also be bacteria cells have an overproduction of harmful proteins because of that energy?
Bacteria have an overproduction of harmful harmful to whom?
I'm not sure, but isn't that because of that energy? I'm sorry, never mind.
But I'll go with that because I let me see if we get there eventually. But this idea that if you express all of the genes all the time, that would be somehow harmful.
Yes.
Yeah? How could it be harmful? I want to work with that argument to everybody. So how could expressing all the genes at once be harmful?
Can it have something to do with their life cycle? So like if bacteria is not infecting something at that moment, they possibly wouldn't be expressing
certain factors, but if they express them all the time,
wouldn't it be detrimental because they're wasting more resources on stuff that might not necessarily be
so again, we have these two competing ideas. So that's more along the lines of it's not economical, it's wasteful. And anything that is wasteful in the longer run might be selected against. But then there is another idea here that it not only is it wasteful, something that you'd like to avoid if you can, but it's also harmful, toxic. If you express all the genes at once, it might be a little too toxic. And there was this interesting idea here that if a bacterium is not at a certain type of their life cycle, why express the things that it might need at those stages if they're not there yet? So I'm gonna go with that, and it's a way linked to this idea of toxicity for expressing everything. It kind of goes along with that concept, the concept that bacteria and cells in multicellular organisms have the genes that they need under different circumstances. But if you express all of the genes they have under one circumstance, you may start having competing interests within that bacterium. So one example that I like to use is bacteria may express certain genes that have to do with extracting energy from nutrients. But they may also have genes that have to do with using nutrients for building biomass. So the same, we sort of talked about this before, the same ATP molecule could be used to extract energy and move things around or generate a bunch of catalytic reactions that require energy. It can also be used to make new RNA molecules, right? And a closely related molecule, DATP, can be used to extract energy, or it can be used to build DNA. So at times there may be these conflicting decisions that bacteria have to make that is not only wasteful to run them all at once, but it can also be a little counterproductive, because I'm building something but I'm destroying it. Right? I'm putting this thing here, but then I'm taking it away. So often, if bacteria were to express all of their functions simultaneously, in essence, they would stall. They wouldn't really be doing anything. They wouldn't be dividing. They wouldn't be consuming energy. They wouldn't be infecting. They wouldn't be doing what they need to do. Okay? So even in unicellular organisms, very simple ones like bacteria, there is a certain level of making decisions. And that's what gene expression regulation is about. Deciding on what genes to use at any given time of all the ones they have, based on those environmental conditions, based on those situations they find themselves in. Okay? Cool. So all cells decide which genes to express. So that's an on or off decision. Sometimes they decide how much they will express of them. So often it's not only expressing a gene or not, but also if they're being expressed, how much they're being expressed. And cells make these decisions based on intrinsic and or extrinsic signals, sometimes a combination of both, right? And notice how I'm saying all cells here. I'm not even going into prokaryotes, eukaryotes, because I really mean it. It's all cells. Both prokaryotes and eukaryotes do these things. And the whole collection of molecular mechanisms by which cells carry out these decisions is what we collectively refer to as gene expression regulation. Okay? Yes?
Just one quick question.
Uh-huh.
Last slide, you said all bacteria, I think, but bacteria, are you saying like one population of bacteria?
I mean a species. In a given species, all of the cells of a bacterial species, E. coli, you would expect that they're all the same. So why aren't they just expressing all the same genes? Well, because if they were expressing all of the genes they have, with multicellular organisms, there is the argument that if a muscle cell starts expressing intestinal cell genes or neuron genes, it can't be a muscle cell, right? Because it has neuronal proteins and intestinal cell proteins, and it can function. With bacteria, why not have a minimum set of genes and express them all the time? And now we've gone through two strong arguments for why that might be disadvantageous. Yeah? But that's what I meant by bacteria are all the same within a species. Yeah? Cool. So when we start looking at the details, that's when we start seeing some differences between prokaryotes and eukaryotes. So you've seen this before, left side bacteria, right side eukaryotes. And when we look at the different mechanisms for GER in these groups, we notice that there are two that are shared by both, transcriptional regulation and post-translational regulation. What that means is both bacteria and eukaryotes like to regulate when and how much a gene they will transcribe or not. And they also like to regulate the activity of proteins after they've translated them through mechanisms that we kind of hinted at a couple lectures ago with these chemical modifications after a protein has been translated that can activate a protein or deactivate it or change its function somehow. And that we'll discuss in more detail next week. Notice that in eukaryotes, there is a whole bunch of extra regulatory mechanisms that don't exist in prokaryotes, partly because of what eukaryotes do to their protein coding mRNAs. There are certain things that eukaryotes do to their mRNAs that prokaryotes don't, and each of those things that eukaryotes do open up opportunities for control points, for regulatory points, right? And the other thing to notice about this slide is that it's very natural, very understandable that we would gravitate to thinking that regulating gene expression is regulating gene transcription. And I'm not saying that's not a very important part of gene expression regulation. It is, but it's not the only part. So there are mechanisms that have to do with when a certain mRNA is translated or what cells do to those proteins after they've been translated that also contribute to gene expression regulation. And this is because when we think about what expressing a gene means, we need to take a step back and understand that expressing a gene is, for the cell, and by extension the organism, experiencing that phenotypic function that the gene brings, right? If I say a gene is a collection of letters in DNA, well, those letters in DNA don't do anything. They don't move things around. They don't carry out chemical reactions. They don't really do anything. They carry information that is used to make an RNA. Those RNA molecules don't do much either by themselves. Right? In many cases, they need to be translated into a protein, and that protein often needs to be post-translationally modified to finally carry out a function, such that that gene is expressed phenotypically, right? That there is some sort of physical consequence of having that gene. Until then, all of these mechanisms will contribute to whether or not those genes are expressed, and therefore we include them as mechanisms of gene expression regulation. Okay? So in most textbooks, you'll see that there is, in eukaryotes, 4 stages of gene expression regulation: transcriptional, post-transcriptional, what we do or what cells do to RNAs after the RNA has been transcribed; translational, when an mRNA is translated or not; and post-translational. Some textbooks include a prior stage that we'll discuss in more detail next week, that we could call pre-transcriptional regulation or epigenetic regulation, and we'll discuss that on Tuesday. And that is the one that has to do with DNA accessibility. So not only do cells decide when to transcribe certain regions or not, but also they make decisions as to what areas of DNA should be accessible or not. Okay? Okay. So the rest of today is about GER in bacteria, in prokaryotes. Bacteria are all about economy. And what they have become throughout evolution is super efficient gene expressors by expressing only the genes they need and expressing groups of genes that normally function together in a combo or a packet, right? So you may have heard this before, and if not, I'll introduce the concept. Bacteria organize many of their genes in what are called operons. These are groups of genes that are transcribed together and translated together such that when they're needed as a group, they're all expressed together. When they're not, they're not. Right? So there's no waste there. Everything has become much more efficient. Now, we'll talk about the operons and how they're turned on and off in a few slides. But before we go there, I wanted to go through some of the very basic and general aspects of transcriptional regulation and emphasize the fact that transcriptional regulation is an interplay between 2 things. One is sequences in DNA. These sequences carry, as we will see in a couple of slides, they carry the regulatory logic behind the expression of a given gene, in the sense that it is through those sequences in DNA that evolution wrote these genes should be expressed under this and that other condition. Okay? So those are sequences in DNA that belong physically in the regulatory promoters or regulatory regions of genes, right? Or in the case of bacteria, sometimes in operons. Then there is the DNA binding proteins. If the sequences in DNA are the encoders of information, the DNA binding proteins are the interpreters of that information. Those are the entities in cells that actually do the reading of this regulatory logic, right? So the way this works is these DNA binding proteins are divided between activators and repressors. Some of them will turn on transcription, some of them will inhibit or repress it altogether, right? And they bind to these sequences in DNA to activate or inhibit the recruitment of RNA polymerase to the core promoter of a gene or an operon. So mechanistically speaking, there is certain sequences in DNA that can be bound by activators or repressors, and those will in turn accelerate or go against the recruitment of RNA polymerase to the core promoter of those genes. Right? Yes?
So this is pre-transcription?
It is transcriptional because what the cells are doing right now is deciding whether or not they will transcribe a gene. Right? When in eukaryotic cells we talk about pre-transcription, is when we basically cells decide if those sequences in DNA will even be visible to the cell. Right? A little bit of nomenclature, and this is something that was borrowed from old days in chemistry. Sometimes you'll see references to sequences in DNA that regulate the expression of genes they're associated with as cis regulators. And the idea here is that those DNA sequences are part of the same molecule that contains, or part of the same molecule they regulate, right? So I don't know who came up with this really, but this idea borrowed from chemistry, this idea that anything that is cis is on the same side of a molecule versus anything that is trans. So sometimes you'll see these DNA binding proteins referred to as trans regulators of gene expression. So cis regulators and trans regulators. Yeah. That's all nomenclature, but you still see it in the literature, books, papers, talks, and whatnot. Luckily, now things have become a little more explicit, and we talk about transcriptional regulators for those activators and repressors, and we talk about sequences in DNA that bind those regulators. Any questions, anyone? Yeah.
Just clarifying, cis is for the sequences and trans is for the proteins.
Yes. All right. So let's talk about these transcriptional regulators, both activators and repressors. They generally, not always, but generally have three key domains, three different regions of those proteins that carry out specific functions. So first is the DNA binding domain. These are proteins that interact with DNA, and they need those DNA binding domains. These are collections of amino acid residues that interact with a particular sequence of nucleotides in DNA. So this is not, these analogies, it's a little anthropocentric, but it's not completely wrong to think about these proteins being able to read DNA sequences. The way they read DNA sequences is not because they can understand the collection of letters in a region of DNA, but because there is a certain combination of amino acids in their DNA binding domains that interacts molecularly, biochemically, with very specific collection of letters in DNA, right? So sometimes we say transcription regulators can read DNA. A more appropriate metaphor might be that these transcriptional regulators can identify certain words in DNA and latch onto them. So they don't know how to read DNA, but they do recognize certain words when they see them. That's probably a better analogy, okay? The specificity is due to a very unique biochemical fit between a given sequence of nucleotides and amino acids in the DVD, that's short for DNA binding domain. One thing that is not written there, but you'll come across in the literature because it's true, often this interaction between amino acids in the DVD and the corresponding target sequence in DNA is through the major groove of DNA. If you guys remember the DNA helix, the way that the strands are oriented relative to one another, there is an area that is very narrow with a very narrow access to the central base pairs, and then there is the major groove that offers a wider access to those base pairs in the middle. And, it stands to reason that proteins would use that wider access to access DNA sequences. Then another thing that all of these activators and repressors will have is a domain for transactivation or transrepression, right? So it's one thing to bind to DNA at very specific locations through this interaction between the DVD and the target sequence, but then they need to do the thing they do, either promote binding of RNA polymerase and transcription or inhibit it. And they do that through these transactivation or transrepressor domains. Now, how these domains work differs. In some cases, is simply the facilitation of the recruitment of RNA polymerase via protein-protein interactions. So a transactivator may bring RNA polymerase to the core promoter because it has molecular affinity with it. And whenever RNA polymerase sees these proteins in DNA, it likes it there, gets close, and then it finds the core promoter. Other times it's a little more indirect. These transactivator domains do things to DNA. This is a little more common in eukaryotes. These transactivator domains will do things to DNA and leave certain marks in DNA that make now those regions of DNA more desirable for the transcriptional machinery, the transcription factors that form the initiation complex, RNA polymerase, and the like. Right? So sometimes it's a direct interaction with RNA polymerase or the transcriptional machinery that brings these things to a specific gene. Sometimes it's a little more indirect. I leave a mark, and then I let the machine take it from there. And I mentioned here 3 key domains. There's a third one that not all of them will have, but they do more often than not, I suppose. And that is the domain that these activators and repressors use to carry out that logic, that regulatory logic that we were mentioning before, right? So cells can sense signals from the internal environment and the external environment, and then they use some sort of, I asked for a show of hands earlier in the semester if anyone was from computer science. I don't think there was anyone from computer science, but you guys, have you heard of the term Boolean logic? Some of you have, some of you. So Boolean logic, most of you have experienced it or have seen it before. You may not have known that it's called Boolean logic, but it's the logic that comes in the form of if X and Y, then so and so, right? And we call it Boolean logic because you can start making statements for that if part of the statement that connects conditions. So if A and B, then C, or if A or B, then D and E, and so forth, right? So this Boolean logic is based on if-then statements with a whole bunch of and, or, nor, nan, etc. gates, we call them, right? So these are things that basically integrate information, internal or external, or both, and that allows cells to check certain boxes as to what's going on, and therefore make decisions regarding the expression of certain genes. So it's not crazy to think that what cells are constantly doing is running these if-then statements. They'll start asking questions like, if I'm hungry and I have enough nutrients, then I can express genes that will extract energy out of those nutrients. If I'm full and it's been a while since I divided, but not if it's too hot outside, then, and so forth, right? So it's all this collection of if-then statements with some of these Boolean gates in them. And all of that is carried out by molecules that interact with these activators and repressors and inform these activators and repressors of these external and internal conditions. And when I say these molecules, I really mean it. These are chemicals. These are chemicals that are floating around in cells and have a way to communicate to the cell these type of information. We call those molecules inducers. And sometimes they will activate a certain transcriptional regulator. Sometimes they will turn them off. And they can work both with activators and repressors. So the fact that you see this crazy combination of green arrows and red inhibition bars is to indicate that it could go any of these ways. You could have inducers activating repressors and actively turn things off. You can have inducers inhibiting activators. The effect is the same. You're going to be turning something off, or the other way around, right? So there's all kinds of combinations that can take place. Here in this diagram, the yellow circle points to how small these inducer molecules tend to be and how small these allosteric domains are in transcription regulators. So these are regions of the protein that serve as a landing pad or a pocket for these inducer molecules. So when the inducer molecule fits in those pockets, they will induce, I'm repeating myself, but that's the terminology, but they will trigger a series of conformational changes in the rest of the protein that will have an effect on what that protein does. So for instance, you can have an inducer that binds to the pocket of a transcriptional activator that was inactive. It was not binding to DNA, right? And it was not binding to DNA because its shape was such that its DNA binding domain was not in the proper conformation to read the usual word that they would detect in DNA to bind DNA, right? But then the inducer comes along and triggers a conformational change in that protein such that now that DNA binding domain changes its shape a little bit and acquires the shape that is perfect for it to recognize certain combination of nucleotides in DNA. And now, anytime that transcriptional activator sees those combination of nucleotides in DNA, will be able to bind to it and stick to DNA in that particular location and do what activators do best, which is recruit RNA polymerase and induce the expression of a nearby gene. That makes sense? So these inducers will change the shape of these transcriptional regulators and make them better at what they do or worse at what they do. And that's how they regulate the activity of these transcriptional regulators and indirectly execute regulatory logic. Although that was super vague, now we're going to go through a series of examples and put these in practice. So we'll talk about two operants. These are the two most famous. They're in every textbook, every video, and whatnot. We'll start with the simpler of them. There is the TRP operon, short for tryptophan operon. And let's start with the operon itself. So this is an operon that codes for 5 enzymes that are critical for making tryptophan in bacterial cells. Tryptophan is one of the amino acids bacteria need to make tryptophan to express their proteins, and they use these 5 enzymes working more or less in concert, right? Working as a part of a pathway like those that we had seen with Biddle and Tatum to make tryptophan out of precursor molecules, right? So the regulatory logic behind the synthesis of these enzymes and the synthesis of tryptophan goes something like this. Bacterial cells need tryptophan, and they're constantly making tryptophan, but they're also constantly using that tryptophan to make proteins. Now, there may be certain circumstances under which the bacteria or bacterium may have made a little too much tryptophan. So here, if the cell has made too much tryptophan, then save energy by turning this thing off. So under regular circumstances, when bacteria need tryptophan, they make it by use it. They consume what they produce. The operon is on. RNA polymerase is constantly transcribing through this operon. This will give rise to one of those polycistronic mRNAs that we discussed before. It's one long mRNA molecule that encodes 5 different proteins in it. When that mRNA molecule gets translated into these 5 different proteins, those 5 different enzymes will make tryptophan, right? But if there is too much tryptophan, why bother? And the operand turns off. So my question to you, what inducer molecule could communicate to the cell that the cell has already too much tryptophan? If you were to design this, what molecule would you use to tell the cell, hey, you have too much tryptophan? A repressor? The repressor is what you would use to turn it off. But the question I'm asking is different. How do you tell the cell there's too much tryptophan? Tryptophan itself, right? Like all of those negative feedback mechanisms that you've probably seen before. The end product of something is what ends up turning off the process that leads to it, right? It's the most direct way of control. If this whole thing makes tryptophan and the bacterium is making a little too much tryptophan, how can it know? Well, based on tryptophan, right? It's the simplest. And that's exactly what happens. So what happens is, when there is way too much tryptophan, there's a protein, a repressor, that is floating around in the cytoplasm that is normally empty, right? So the allosteric domain for that repressor protein is normally empty. Why? Because that allosteric domain normally binds tryptophan, but the bacteria is busy using that tryptophan for everything else. It's using the tryptophan to translate all kinds of different proteins. So there is usually a steady state but low level of tryptophan floating around unused. When for whatever reason tryptophan starts accumulating, some of those molecules will bind the allosteric domain of the repressor. And when that happens, the repressor that is now occupied with its inducer tryptophan molecule, and when that molecular conformational change, and it can now detect its target DNA sequence in DNA. Right? So the shape of the repressor changed such that now it can bind DNA. And it binds DNA in a region that we call the operator, where the binding sequence for this repressor lives. And the operator happens to be, this is not by chance, this is by evolution, happens to be located between the core promoter and the rest of the operand. So what happens is physically, now this repressor is blocking RNA polymerase. It put up a physical barrier to RNA polymerase, and RNA polymerase can't get through, and the whole operand stops being transcribed. No more enzymes to make tryptophan. There's a lot of tryptophan in the cell, but eventually that tryptophan will be used up, right? So when that happens and the levels of tryptophan drop again, that tryptophan that was bound to the allosteric domain in the repressor protein may decide that it wants to leave. All of these things, if you notice, all of these binding of inducers to transcription regulators is not permanent. It's a dynamic process. So the more molecules of the inducer there are in a cell, the higher the chances they might find that allosteric domain unbinded. But they will dynamically be coming off every once in a while of those molecules. If there's still a lot of tryptophan, another molecule will come in and occupy its place. But if now the cell is kind of empty, the chances that a new tryptophan molecule might come and take its place are lower. The repressor empties up, and now that it's empty, it can no longer bind to the operator sequence because it changes shape, and it no longer has that DNA binding domain in the perfect conformation to bind the operator sequence. It falls off from DNA, and now RNA polymerase can get through the operator and transcribe the operand. Yes?
I'm sorry, is this a transcription factor? No.
The one that is a transcription factor is, okay, careful with the nomenclature. So the repressor we call a transcriptional regulator. A transcription factor in bacteria, you could say that a regulator is a transcription factor as well, but in eukaryotes it gets a little trickier because transcription factors were those that don't regulate anything, don't make any regulatory decisions. They're the TF2 family of proteins that binds the core promoter and recruits RNA polymerase. Those are, it's in the name, transcription factor 2. All these other guys, we call them trip repressor, lac repressor, this repressor, that activator, and so forth. They have those names because they just regulate the expression of these genes.
So this is only in bacteria then?
So far, yes. Everything that we've been discussing about the trip operon is in bacteria. Yeah. Okay. So we went through some of this using the trip operon as an example, but let's get a little deeper into definitions. There's two situations that can occur for operons. The one that we just described is here on the right. The operon is normally on, expressing the genes that the bacteria needs, but every once in a while it turns off under extreme or less frequent circumstances, right? In the case of the trip operon, that less common circumstance is having a lot of tryptophan laying around. That's not common for bacteria, right? So when an operon is normally on, but every once in a while can turn off, we call those operons repressible. The operon is being repressed. Now, there are many other operons that are the exact opposite. They're normally off because there is something that the bacterium normally doesn't encounter, but it has the tools to maybe deal with a situation that happens once in a blue moon, but when it happens, it wants to be ready for it, right? So there are operons that are normally off, but under certain rare circumstances will turn on. And those operons we call inducible. So there is this layer of definition or classification of operons between inducible and repressible. And it has to do with what is the common state of the operon and what is the rare state of the operon. Then there is this other layer of classification. And it has to do with what type of transcriptional regulator does the switching, right? So for the trip operon, we just saw it was a transcriptional repressor. There are other operons in which the protein doing the switching is a transcriptional activator, right? So depending on who's doing the job, we call operons under positive regulation or under negative regulation. Now, here's where it gets a little complicated. It turns out that these two layers are independent from one another. What that means is that you can have all possible combinations. You can have let's start here in the bottom left. You can have inducible operons that are under positive control. What that means is that they're normally off and something, a signal, tells them you should turn on. And the way that that switch is carried out is through a transcriptional activator. So what the inducer is doing there is facilitating the binding of an activator. The operon is off because the activator is not bound to DNA. But a signal, a rare signal, tells this activator, you should go bind DNA. And now the operon is on. So because it went from a common off state to a rare on state, we call it inducible. And because the protein doing the switching is an activator, we call it under positive regulation. Inducible under positive regulation. Let's look at the bottom right now. An operon that is inducible, but under negative regulation. So the operon is normally off. And I forgot to add that. I apologize. It's a good thing that I realize now. There you go. It's off because there is a repressor binding to it under normal circumstances. But then a signal comes along that says, you know what, we should turn this thing on. So how do you turn something on when it's off because of the presence of a repressor? Well, basically you take that repressor out. So in this case, what the inducer will do is take the repressor out of DNA. And now without that repressor, the operand turns on. If you understood those two, kind of easy to understand these two repressible operands on top, right? So a repressible positive is one in which an activator normally present in an operand, keeping the operand on, is taken out by the signal, so the operand is being turned off, hence repressible, and it's under positive regulation because the protein doing the switching is an activator. Or you can have a repressible negative operand where the operand is normally on because there's a repressor that is floating around the cytoplasm not doing anything, but then a signal tells the repressor, you should go bind DNA. Yes?
So for the inducible, so if the repressor is taken off, does an activator get bound to it?
Not always. Sometimes just taking the repressor, that's a good question. So the question is, I'm going to repeat it for everybody. So the question is, in an inducible negative, when you take out that repressor, do you need an activator to come in and turn on the... Not always. Sometimes taking the repressor out is enough to let RNA polymerase transcribe through this operand.
So an activator is only necessary if it's like it needs something.
It needs a little extra help, yes. Yeah. That's a good question. Yes?
So there are inducers for all of them, or are they technically repressors on the top two? Right, because you have inducible reactions, which have inducers, and then you have repressible reactions. which also have inducers.
Exactly. So that's the thing. So inducible and repressible has nothing to do with activator or repressor. So the fact that we call it an operand repressible, and trust me, it hurts me more than you because I have to teach it every semester. You learn it only once. A repressible operand can be repressed by an activator. And you're like, what? Well, you can repress an operand by taking an activator out. And then you can induce an operand with a repressor. I'm like, what? Well, you can induce an operand by taking a repressor out. So these two things are independent from one another, and there exist all possible combinations. On that note, we know now that tryptophan is an inducer that will bind to the repressor and shut off the expression of the operand. So what would we call the trip operand? Is it inducible or repressible? No? You didn't say anything? The question is the trip operand, the tryptophan operand, based on what we learned. Is it inducible or repressible? Repressible, right? It's normally on, normally expressing, and a signal turns it off. So it is repressible. And is it under negative or positive regulation? Positive, negative? Negative. Why? Because the repressor is added. So this one is a little easier. For the negative versus positive regulation, just look at the protein. If the protein is a repressor, negative. Activator, positive. That's a little easier. The inducible versus repressible one, you have to sort of understand what is the normal state for a cell versus the induced state or regulated state, right? Okay? Show of hands. For whom is this the very first time that you've seen the tryptophan operand? About half, a third of class. Okay, cool. Has anyone heard of the lactose operand before? Not much more. Okay. Because that's the one that we'll discuss next. The lactose operand or the lac operand. Different operand, so we have to sort of take a step back and go through the regulatory logic again. There is this molecule called lactose. It's a sugar that is made of actually two carbohydrate monosaccharides. So it's a disaccharide made of two sugar molecules. And there is this enzyme called beta-galactosidase that bacteria can use to break that disaccharide into two monosaccharides, both of which are useful, but one of which in particular is super useful, glucose. Why? Because it can use glucose in glycolysis and get started with the whole oxidative respiration and all that, right? So it's a source of energy. Now, here's the thing. Lactose is normally absent in bacteria, right? All of the, let's say, E. coli that we have in our gut now, unless you've drunk a glass of milk within the last, 6 or 8 hours, there's probably little lactose in your gut right now. And that's most of the time for bacteria. So, because lactose is normally absent, the bacteria keep the lac operon, what do you think, on or off? Off. There's no need to, and I should have mentioned that the operon expresses these enzymes that precisely break up lactose. So beta-galactosidase is one of them, coded by the LAC-Z gene. And then these two, LAC-Y brings lactose into the bacterium, and LAC-A will transform lactose into a slightly related molecule that we'll discuss in a second. So the operon is normally off because there's normally nothing to break. What's the point of expressing these things if there's nothing for these enzymes to work on? And the reason this operon is normally off is because there is this repressor that actually binds the operator when it's empty. When its allosteric site is empty. Now, what do you think could be a really good inducer for the lactose operon? What could be a good signal for the bacterium that there is lactose around? Lactose. If you want to tell the bacterium, hey, there is lactose around, use lactose. Right? Pretty straightforward. It turns out that lactose gets converted by that LAC-A enzyme into a very similar molecule, very, very similar molecule called allolactose. And it is allolactose, the one that binds the repressor. So you'll see, like in this diagram, that lactose binds the repressor. To be a little more technical, what binds the repressor is allolactose, but they're almost equivalent. And when allolactose binds the repressor, now the repressor falls off DNA. Why? The binding of this inducer made this repressor change in shape in such a manner that now it doesn't like binding DNA anymore. So with the tryptophan repressor, it was the opposite. The binding of tryptophan made the repressor love DNA, go to DNA and bind DNA. The binding of allolactose here has the opposite effect. It makes the repressor fall out of love with DNA and fall off. Yeah? So now, the presence of lactose takes the repressor out of DNA, and RNA polymerase can read through the operator and the rest of the operon. These enzymes get expressed. The product of the LAC-Y gene, it's a protein called permease that makes the cell permeable to lactose. All of that lactose that was outside, let's say in our gut, goes now inside of the bacterium, and beta-galactosidase, coded by the LAC-Z gene, will now break up that lactose into galactose, and glucose gets used for energy. Yeah?
Where did the allolactose come from?
Comes from lactose itself. What happens is there's a tiny amount of the lactose that is outside that will always kind of leak through into the bacterium, but it's a tiny amount, right? When that tiny amount is recognized by the gene coded by LAC-A, it gets transformed into allolactose. And now it becomes a potent inducer for the repressor. So it's almost like you have a ton of lactose outside. It cannot come in because there's no permease, right? But then one of them kind of leaks through. The cell membranes are never completely impermeable, right? So one of them kind of leaks through, gets converted to allolactose, takes the repressor out, and now there's a whole flood of permease and beta-galactosidase, and together they let all of that lactose in and convert it to glucose for energy.
I thought that LAC-A coded for transcysteine.
That's the enzyme that will convert lactose to allolactose. Yeah.
So the repressor just stops lac Z and lac Y. Like, why is lac A working when the repressor is on?
I was dreading this question. Okay, it's all good. It means that we're elevating the conversation. So the question here is, well, the repressor was stopping lac Z and lac Y. How come it's not stopping A? The answer is that there is always some lac Z and lac Y. Why? Remember what I said about none of these things are covalently bound to DNA? None of these things are chemically permanent. So when the repressor binds to DNA, one slight movement of that DNA molecule and the repressor might fall off momentarily, right? And then until it finds its way back into the operator sequence and binds. So it's all, in a way, statistics. What are the chances that you will find a certain repressor bound to the operator or not? If there's a whole bunch of empty repressors, there's a good chance that those repressors will be occupying the operator. Okay? But every once in a while, a repressor molecule might fall off. For long enough for RNA polymerase to go make 1 or 2 copies of this polycistronic mRNA. You make 1 or 2 copies of beta-galactosidase and permease, and 1 or 2 copies of the transacetylase. Now you let one lactose molecule in that gets converted to allolactose, and that now allolactose will keep the repressor off for much longer. So now you don't make 1 or 2 copies of the mRNA. You make 200 of them, and the thing explodes. Yeah? That's a lot to take in, so make sure that you sleep on this, check the lecture capture. But there is always a little bit of leaky, we call it in jargon, leaky expression of these things. And what these inducers do is sort of lock in certain states. So the lactose turns into allolactose and locks in the on state. Okay? When does this turn off? Right? With the trip operon, it turned off when enough of that tryptophan is consumed. The tryptophan molecule that was bound to the repressor may momentarily fall off of the repressor. If there is a lot of tryptophan, some other molecule will come take its place. Same here. Lactose is bound to the repressor, right? And there's a lot of lactose coming in because permease is being expressed. But you didn't drink a gallon of milk, you just drank a glass. So at some point, the lactose outside will run out. And the lactose inside will be converted to glucose and galactose. So at some point, that bacterium will be empty for lactose again. When that happens, this lactose or allolactose molecule will fall off momentarily, and the chance that another lactose or allolactose molecule will come take its place are very slim now, because there is very little lactose left, right? So when this repressor is empty again, it will go occupy the operator again, and the whole thing turns off again. Until when? Until the next glass of milk you drink. Okay? All right. Again, I'm checking the time. We might have to do it again. It's becoming a pattern. It's just that the conversations are so good. These are good stories. So we're ready to take that quiz after. I heard from some of you that you couldn't take it, despite the fact that I left it open for it. So know that we're going to be taking the quiz sometime before tomorrow, okay? I may leave it open until tomorrow midnight or something, so they have plenty of time. But know that that's happening, okay? So that's not the end of the story with the lac operon. The lac operon has yet another angle to it. And it goes something like this. If lactose is present, sure, the bacterium would like to use that lactose as a source of energy, right? because it can get glucose out of that lactose. But if the lactose is present and that cell is starving, now that bacterium, its life depends on breaking down that lactose. Does that make sense? It's one thing to say, there's lactose. Yeah, sure, I could munch on some lactose. It's another thing to say, lactose is the only energy source that I have around. So now I better make sure that I use it. And that regulatory layer exists. So what happens is, when lactose is present, based on everything that we just discussed, allolactose will lift the repressor off of DNA, and RNA polymerase can get through that operator and transcribe the operon. If lactose is present and the cell is starving, then not only will RNA polymerase transcribe through that operon, but it will also transcribe much more than it normally would. There is an activation of transcription of this operon because now the bacterium's life depends on it. Okay? So, same question that I've asked before a number of times, this time it's a lot less obvious. So what molecule tells the bacterium that it's starving? How do you signal hunger? ATP. But ATP means energy, right? So how do you use ATP to signal starvation or hunger?
After ATP is converted.
So exactly. So because ATP is the currency of the cell, is the molecule that signals energy, one of the easiest ways to tell a cell, buddy, you're starving, is to let the cell know that there is little to no ATP. And that's exactly what bacteria do. They let the cell know that they're starving because of how little ATP there is. So the way this works is, when glucose is high and there is plenty of ATP, the cell sure might use some lactose, but doesn't really need it. On the other hand, when glucose is low and there's very little new ATP being made, remember, if you guys remember your glycolysis and oxidative phosphorylation days, glucose gets transformed into a bunch of molecules that through the Krebs cycle and the electron transport chain get turned into 38 ATP molecules, remember? All right. So when glucose is low, there is no new ATP being made, and whatever there was is consumed. Right? So when glucose runs out, it's just a matter of time until ATP runs out as well. Now, ATP is not destroyed. What happens to ATP when the cell needs energy is the cell will take out the phosphate groups from ATP. Sometimes it will turn an ATP into an ADP. Sometimes it will turn into an AMP for adenosyl monophosphate. Right? And it turns out that AMP can be turned into a basically almost identical molecule called cyclic AMP. It's just a little intramolecular bond that AMP goes through, and it turns into cyclic AMP or CAMP. That's done by an enzyme called adenylyl cyclase. Okay? And it turns out that cyclic AMP, it's a really potent inducer that tells the cell, or that the cell uses to inform itself that it's running low on energy and that it's kind of starving a little bit. And the way cyclic AMP does that is by binding an activator protein, a transcriptional activator called cyclic AMP activated protein, or CAP. So you guys remember minutes ago we were talking about the tryptophan repressor protein and the lactose repressor protein? This time we're welcoming an activator protein. This activator protein is normally off of DNA because it's not needed in DNA. But when there is a lot of cyclic AMP bound to CAP, now CAP changes conformation and loves its target sequence in DNA. And if it finds them, it will bind to them. And this CAP protein has the ability to recruit a lot more RNA polymerase to the promoter of the lactose operand, right? So if the cell is starving and there is a lot of cyclic AMP will bind to cap, and the complex cyclic AMP cap will bind DNA and start bringing in a lot more RNA polymerase that normally would come to that operon, and all that RNA polymerase will transcribe a lot of this operon. Why does it transcribe a lot of this operon? Because we're still under the scenario in which there is some lactose around. And because there is some lactose around, the repressor left the operator sequence. The operator sequence is empty, and RNA polymerase, all of those RNA polymerase molecules can get through and transcribe this operon. Not once or twice, but hundreds of times. So now the bacterium makes sure that it has the enzymes that it needs to consume possibly the only source of energy it has. Making it a little more dramatic than it should, but that's more or less the evolutionary logic behind this. Yeah? Questions, anyone? So far, so good? Yeah? Okay. So now, knowing what you know, this is one of those group activities. I'll give you a couple minutes. Where would you place the lac operon in a little space of inducible versus repressible, positive versus negative? All right, so what are we thinking? Where does the lac operon go? Which way? It's only 1 out of 4, so let's put on our betting hats. Inducible negative. I hear some inducible negative, some heads nodding along. Okay, so why inducible?
It's normally off when it's being turned on.
And why negative?
There's a repressor.
Because the one doing the job is a repressor, right? So inducible negative. So lac operon here. Does anyone disagree? You're saying there is a repressor, but yeah.
Isn't it inducible positive?
Why?
Because it needs CAMP to bind to cap, and cap produces.
More, right? So is it?
It's not that it's at like low levels, it's starving.
So is it the repressor doing the job or the cap protein doing the job? The activator. Right? Ha! So, ding, ding.
Yeah, is it just
Can it be both because technically it's two separate operations?
Oh, you're forcing me to do this now? Oh my God. Somewhere in the, both in the middle. That's what I should do. Check this out. Oh my God, look at that. I'm joking. No, it's actually inducible negative. Let's erase that. And here's why. Oops. It's inducible negative because the presence, absence of lactose is what drives this operon. The cap cyclic AMP thing is just a modulator. But the switch, the on-off switch, is the presence or absence of lactose. Another way of saying that is the energetic status cannot trump the regulation by lactose. In other words, you can have the cell starving to death. If there is no lactose, that thing will stay off. Because if the cell is starving and there is no lactose and the cell starts producing these enzymes for breaking down lactose, it's making bad matters worse. It's starving and it's wasting energy in enzymes that can't use anyway, right? So the presence of lactose, or absence of lactose, the presence-absence of lactose kind of tramples the energy, low energy, high energy status. So ultimately, this operon is turned on by a signal, the presence of lactose, making it inducible, and that switch is a repressor, making it under negative regulation. The cyclic AMP cap thing matters, but it's just a final tuning after the fact. Okay?